Hi, I'm Mr. Williamson. In this video, we're going to look at the ways cells maintain homeostasis. First of all, homeostasis is the act of the cell remaining the same, keeping things consistent inside the cell, whether we're talking about the salinity, the osmoregulation or water regulation, or whatever. How does the cell maintain homeostasis? How does it get things into and out of the cell? Let's get started. So, first of all, you need to remember that the cell's unit is a unit of structure. It's the smallest unit of life. There's nothing smaller that maintains all the characteristics of life. It, for instance, can reproduce. It responds to stimuli. It grows. It reproduces. Okay, its internal environment is maintained by the cell membrane. All right, it's maintained by this structure right here, this line, this boundary. You have outside the cell and you have inside the cell. And that membrane maintains what is in and what is out. All right? And it controls the entrance and exit of the things to and from the cell. As a quick review, we've gone over this before. Remember that the plasma membrane, or the cell membrane, is a fluid mosaic. Mosaic meaning it's made up of a lot of little parts, and fluid means that it is moving. It is not locked in place. It is selectively permeable, selectively semi-permeable, meaning that it can actually determine in a lot of times what goes through it and what does not. All right? And it is composed of phospholipids, proteins, and other substances. Probably the most important ones we want to talk about are the phospholipids and the proteins. Remember the phospholipid layer. And again, you don't need to know the structure of a phospholipid. Just get the idea that you have a hydrophilic head, all right, which is attracted to H2O, water. And then you have the tail, which is repelled by the water, the two tails. All right, so you have the head and you have the tails. And as a result, we get this bilipid layer. One layer of lipids and then another layer of lipids. Bilipid layer. All right? Transport across the membrane, the most obvious way that we've talked about before in the past is diffusion. And diffusion is just the movement of a substance from a high concentration to a low concentration. So here you can see on one side of the membrane we have a lot of green spheres, and on the other side we have a lot of red spheres. Through diffusion, they are going from a high concentration to a low concentration, so the red ones move this way, the green ones move this way, until hopefully equilibrium is reached. Equilibrium is where they're spread equally and evenly across both sides of the membrane. This does not require energy. The cell has to spend no energy for this to happen. It happens naturally because of the concentration gradient going from a high concentration to a low concentration. Okay, one of the forms of that is osmosis. I have no idea why that letter is blue. Anyways, osmosis is the diffusion of water across the membrane. You have to have the membrane because that membrane is what holds back the solute, the thing that the water is dissolved. So in the the picture here, you can see that in this U-shaped tube, and the barrier's right there, all right? In the U-shaped tube, we can see a high concentration of solute or low concentration of water. They're related. If you have a high concentration of solute, as we learned in the gizmo, the concentration of water is low. If you have a low concentration of the solute, the thing that's being dissolved, you have a high concentration of water. And so you have a higher concentration of solute on this side and a lower concentration over here. So which way is the water going to go? It's going to go from the high concentration of water to the low concentration of water. It's going to go this way. And you can see that in the square down here, or the rectangle down here. The water is going to move across the membrane from the high concentration of water to the low concentration of water. And as a result, because water is moving across the membrane, it's going to push that water column up higher. And I'm going to show you an example of that here in a minute. We talked about, again, don't know what's going on with this stuff up here. The colored letter, I don't get that. We talked about an isotonic solution where the water, the concentration of water and or solute is the same inside the cell as it is outside the cell. I'm going to continue talking about the concentration of water. So the concentration of water inside the cell and outside the cell is equal. Therefore, you have water going into the cell and you have water coming out of the cell at the same rate. Same thing in the animal cells. The result, there should be no change in the cell. You have a hypotonic solution where the concentration of water is greater outside the cell than it is inside the cell. Therefore, the water would enter into the cell, and if you are putting more water into the cell than is coming out, the cell would get larger and could actually lyse. Okay, in an animal cell, and you can see that the example here, in the animal cell, it could actually explode. In a plant cell, because of the cell wall, it becomes very, very stiff. That's what why the grocery stores put water on their vegetables to keep them turgid, meaning the cells are full of water and it keeps them stiff and not wilted. All right. And then finally, we have a hypertonic solution where the concentration is greater inside the cell than outside the cell. The water would leave the cell going to the environment, and that would cause the cell to shrivel up or plasmolyze. And as you can see, I've shown you examples of this in a plant cell where the cell membrane actually pulls away from the cell wall and shrinks. All right, so I'm going to give you an example of this. I actually did this at home and recorded it. And hopefully, if it works out, I can add some of that to the video. But basically what I did was I started with a regular egg, except the one I actually used for my experiment was white. And the whole process, I mean, you're going to see this in the video, but the whole process is basically, and we do this in class, and it's a great example of osmosis. We soak the egg in vinegar, which removes the shell. Now, the whole idea of an egg shell is, if you think about it, when the egg is laid, everything for that chick, that bird, has to be inside that shell or be able to get through the shell. All right, so what do you need for life? Well, you need nutrients. That's what the yolk is for. The yolk is the nutrients. That's all the nutrients that bird is going to get until it hatches. And by the way, we could be talking about reptiles or amphibians or fish. So, but let's talk about birds, a chicken egg. That chick only gets the nutrients that's in that yolk. It has to develop fully and be ready to hatch by the time that yolk runs out of nutrients. It also needs oxygen. How does oxygen get through? It gets, or into the egg. It goes through the membrane, through the shell. The shell is a protective barrier, but inside of it is a membrane. Okay? And when you soak an egg, shell in vinegar, it removes the shell, but it leaves the membrane. All right? So it kind of becomes kind of squishy. All right? Let's watch this video.
Hey on Sci Guys, Osmosis and Naked Eggs. Welcome to Sci Guys, I'm Adam.
And I'm Ryan.
Today we're going to be exploring osmosis through making naked eggs.
The ingredients you're going to need for this experiment are an egg,
Some vinegar,
corn syrup,
and food coloring.
and the equipment includes a glass
And an old spoon or gravy ladle.
over top of your egg. The vinegar is going to remove the shell off the egg and expose the membrane. The shelled egg will need to sit in the vinegar bath for 24 hours. As our egg bathes in the vinegar, the shell is slowly dissolved. The bubbles clinging to our egg will cause the egg to float, flip, and turn. After 24 hours, you will need to drain the vinegar and replace it with fresh vinegar to continue the process of dissolving the egg's shell.
Now in class,
Let
we
the
do
egg
this
sit in the vinegar
only for
another 24
one day.
hours. After that, the shell
They
will be
do it
fully
for 2
dissolved
days, and that's
the
what I
membrane
did at
will be
home because
exposed.
one day is just
Once the
too
step is
long in the
complete,
classroom.
Pour out your vinegar into a sink, gently catch your egg, and give it a rinse.
Once you have your egg removed from the vinegar, you'll notice it's grown in size compared to this raw egg. This is because water has flown from the vinegar into the egg through osmosis. We're not done yet. The next step of our experiment is to place your naked egg into a glass, gently. Then,
This is the
pour your
part
corn
that
syrup
did not
over
work
top
for
of it.
me because I did not have the second egg.
Bend your old spoon and use it to gently submerge your egg into the syrup. The naked egg needs to sit in the corn syrup for between 24 and 48 hours. The spoon is used to forcibly submerge the naked egg
And if
under
you
the
look
surface of the
carefully,
syrup
you
because
can
the egg will
see
naturally want to float.
the water
If the egg
escaping
floats, The surface of the
along
exposed membrane
the
may
membrane
harden and
of
will
the
also
egg.
cause this stage of our experiment to go much slower. Once this stage is complete, pour out your corn syrup using your hand to gently catch the egg and give it a rinse in the sink.
As you can see, after a couple of days in our corn syrup, our egg is shriveled and small. The previous step, where it absorbed a lot of the water from the vinegar. But we're not done yet.
The next step is to fill a glass of water, add a few drops of food coloring, and give it a good stir.
Now with your colored water, gently place your shriveled egg into the glass of water and leave it for a few more days.
The shriveled egg will need to bathe in our colored water for about 24 hours.
So what
The
you're
egg will begin
seeing
to grow and expand
is
as the water
osmosis.
passes
This would be a
through the membrane
hypotonic
into
solution,
our egg.
and it's going in.
After 24 hours, pour out your water into the sink and gently catch your egg. Let's look at this experiment a little closer. First, we need to look at why the shell of the egg is dissolved by the vinegar. Vinegar is made up of aqueous, or in other words, a water solution of acetic acid, and the egg shell is mostly made up of calcium carbonate. When we place our egg into the vinegar, the molecules in our glass cause a chemical reaction that produces carbon dioxide gas, water, and calcium acetate. Once the shell of our egg is removed and we have fully exposed the membrane under it, something really interesting happens. The water molecules in our vinegar begin to travel through the
So
membrane.
this is
of our
caused
egg
by
into
osmosis,
water.
being in a
The special
hypotonic
thing
solution.
about the egg's
The
membrane
concentration of
is it's
water is
semi-permeable.
greater
What this
outside
means
than
is that
inside. The
only
water
let
moves
specific
in.
molecules pass through it. In this case, the molecules it lets through are water molecules. When we place our swollen egg into the corn syrup, the opposite effect happens.
It's a hypertonic solution. It goes from a high concentration inside the egg to the low concentration outside the egg, pulling the water or moving the water from inside outside, causing it to shrink.
The egg has lots of water molecules and only a few sugars, proteins, and electrolytes. All the molecules want to be in equal proportions on either side of the membrane. But remember, only water is capable of moving through the membrane. So instead, the water moves from where there's a low amount of other molecules to where there's lots. This means that the water leaves the egg and goes into the syrup. As the water molecules leave our egg, it shrivels and shrinks. When we place our shriveled egg into the colored water, there's lots of water molecules, but nothing else outside our egg. Inside the egg, there's almost no water molecules.
This is a hypotonic solution, so the water would be expected to move from a high concentration outside the egg inside to inside the egg, causing the egg to swell.
Which in this case causes the egg to swell. The process of water traveling back and forth.
And you can see the difference in the sizes of the eggs.
Is known as osmosis.
So let's talk about other ways that cells can obtain homeostasis. And in this part, I'm going to talk about facilitated diffusion, active transport, and exo and endocytosis. Obviously, I'm going to explain each of those in farther detail, so I'm not going to do it right this moment. But I'm going to start with facilitated diffusion. Facilitated diffusion is diffusion, moving a substance from a high concentration to a low concentration. But we're going to use this protein channel to help do it. And this is probably because the molecule that we're going to diffuse is probably too big or the wrong type to actually go through the cell membrane. So it can't do that because it's either, like I said, too large, or there are only certain types of molecules that can actually pass through the membrane, and this would not be one of them. So what it'll do is pass through this channel. All right, and there's actually, I'm going to show you a video on this and exactly how it works. You don't have to worry about it, just giving you an example, but it passes through the channel. But it's still going from a high concentration to a low concentration, and it's going to go whichever way. It can go both ways. It can go into the cell or out of the cell, depending on whether the solution is hypotonic or hypertonic. But it's facilitated. I always thought of this as like, if we had the gym full of students and we were trying to have you leave, you're going to leave anyways. You're going out the door, but we're going to have a bunch of people like throwing you out the door, moving you even quicker. All right. You'd get out left on your own means, but by having you pulled out or pushed out the door even faster, we facilitate the diffusion. We speed it up a little bit. All right. That's facilitated diffusion. It looks kind of like this. This is a really quick video.
In the process known as facilitated diffusion,
The guy's not real
a special
excited.
carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process.
Again, notice that it is higher on the outside
concentration gradient
and lower on the inside.
into the cell
So it's
where
going to go
released.
with the concentration gradient.
Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration.
The attachment of the molecule to the protein causes the protein to change shape, allowing the molecule to pass through.
Facilitated diffusion
When equilibrium is reached, it no longer takes place.
facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport
Again,
the molecule
the high would be
out of the
down
cell.
here. Second way of doing this is active transport. The word active should imply there's a use of energy, and that's exactly what it is. We're talking about using ATP for energy, for diffusion. And it's usually we're going to go from a low concentration and we're going to travel to a high concentration. From a low concentration on one side of the membrane over to a high concentration. All right? We're going against the concentration gradient. All right? Against the concentration gradient. And the way this works, and again, you just need to understand the big part for this, you need to understand this requires energy. It goes across the concentration gradient. All right? It's kind of like if everyone's leaving the gym and you're trying to get back in, you're going to fight your way in against all these people leaving. That requires energy, okay? All right, so you can see here you have the solute, which this is the low concentration, and this is the high concentration. All right, we're going to go the opposite way. So, again, it has to do with the proteins and the shapes of the solute, but they would bind into that protein, which would then, as you can see, and then we need energy. The phosphate is broken off, which we're going to talk about next unit. The energy is released, changing the shape of the protein, and those two are released back into the higher concentration. All right, and then the last picture is just showing you that the phosphate is released that can be reused in making ATP, which we'll talk about later. Okay, again, showing you one more time, where we have a high concentration of the solute, we have a low concentration of solute, and we're going to move it this way. The solute, the thing we want to transfer, the thing we want to diffuse or move across the cell membrane, is going to move into the protein. We take an ATP and break off a phosphate, which again, you do not need to know that. You'll know it next unit. That releases energy. When the energy is applied to the protein changes shape, just like a clamshell actually. It kind of goes like this, and we release the solute into the high concentration area. Right, but it does require energy. This is what happens a lot in your nerve cells. And if you take anatomy and physiology, you'll learn that. All right, and then there's the endocytosis and exocytosis. This is the transport of large molecules, maybe even other organisms and things like that. Endo means, the prefix endo means in, and exo means out. And over in the picture, you can see that you have the cell membrane. This is the cell membrane here. You have the particles, whatever it is that's going to be brought into the cell. They become, the membrane makes a little fold or a pocket, and it brings in some of those particles, whatever those particles may be, okay? And then it continues to fold that pocket in until it seals it off, and then that membrane, the cell membrane, actually becomes the vesicle, the ball of membrane containing the substance that was brought in. That is called phagocytosis. The prefix phagio or phago is to eat, cytosis of the cell. So this is the cell eating something. I'm going to show you a video on that in a minute. And then there's pinocytosis, which is the cell bringing in the liquid, usually water. All right. In exocytosis, we do the exact same thing. We do the exact same thing, but we do it in reverse. You start with a vesicle, probably, let's say, from maybe the Golgi body that has made enzymes or proteins for outside the cell on the ribosomes. They move until they actually touch the cell membrane, and then they form and combine with it because they're both made of the same phospholipid bilayer. When that happens, the exact opposite happens. It pops. It's like a bubble popping, and those substances are released out into the outer part of the outer environment of the cell. All right. It's almost exactly like I just described. It would be like if you were a bubble popping. Picture a bubble rising up from the below the surface of the water. It reaches the surface, and then it pops, and it releases the gas that was inside of it. That would be exocytosis. Endocytosis, you've never seen because bubbles don't do this, but it would be if a bubble formed around a gas and then brought it back underwater. Here is a animated video of that. It's actually pretty good. I think it's real short. So,
exocytosis is another method of vesicular transport that moves large volumes of fluid or chemicals out of the cell. Vesicles, enclosing substances for release,
and you can see this is supposed to be the cell membrane.
Seal to plasma membranes, and the contents are exported out of the cell.
And this could be whatever waste or enzymes or proteins that the cell is trying to get rid of from inside the cell to outside the cell. Like a bubble popping on the surface of the water. Another video, the same thing.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross
and you can
the
see a
plasma membrane.
bacterial cell about to be engulfed by probably a white blood cell.
In this process, the plasma membrane extends outward and surrounds the food particles.
Goes out and it forms around the bacteria.
Major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis.
Don't worry about those.
The material the cell takes in is particulate, such as a bacterium or a fragment of organic matter. The process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
And finally, this is actually an amoeba eating a paramecium, and you can, we watch this, here's what you're going to be looking at. This part right here is an amoeba. An amoeba is a single cell organism that basically it is kind of free form. It has these things called pseudopods. This is a pseudopod, and that is a pseudopod. The word pseudopod starts with a P. And you will see cytoplasmic streaming. You'll see the movement of the cytoplasm in this area when I start the video. And it will basically, it can turn that part into a liquid for us, essentially, and it will continue to move down, and then when it forms the shape it wants, it actually hardens and becomes hard. And it's going to surround this paramecium. A paramecium is also a single cell organism that is the food of the amoeba. So what you're going to see is that. So you can see the paramecium forming the pseudopods around the paramecium, the amoeba forming the pseudopods around the paramecium, and it will. The paramecium can't escape because probably because it's under a microscope slide and there's glass above and below. But you'll also see the amoeba start on over in this area right here. Not that area right there. Over on the top left, right next to the paramecium, it's probably starting to put the pseudopod above and below, and it will eventually fill that all in. All right. So, real quick review. We talked about the way cells maintain homeostasis. Homeostasis is the cell maintaining itself, whether it's water or nutrients or whatever. We also looked at the way cells bring in things into the cell and get rid of things. So we talked about diffusion, which we've already talked about before, the movement of a substance from a high concentration to a low concentration. We talked about osmosis, again, the movement of water through a membrane from a high concentration to low concentration. We talked about facilitated diffusion, where proteins in the cell membrane are used to help the diffusion from a high concentration to low concentration. And it takes special channel proteins to allow that to happen. We talked about active transport, where we're actually going from a low concentration to a high concentration, which takes energy. The cell has to actually use energy to do that. Because we're going from a low to a high concentration. And then finally, we talked about endocytosis and exocytosis. Endocytosis is the cell going out and surrounding, essentially, a substance or something that it wants to bring into it, and doing that with the cell membrane. Because it's a fluid mosaic cell membrane, it's able to move around the substance or object and bring it in. Or exocytosis, where you have a vesicle, again made out of cell membrane, surrounded by the cell membrane, which goes up and, like a bubble in the water, joins the cell membrane, pops, and that cell membrane actually becomes part of the cell membrane. Hopefully you took good notes. If you have any questions, make sure you ask an Ed Puzzle. Talk to you later. Good luck.